Welcome to the Hope and Focus podcast, where hope has a voice. We highlight the journeys of those impacted by Leber congenital amaurosis and other inherited retinal diseases, and the work driving change. Thanks for being here.
Welcome, everyone, to the Hope and Focus podcast. I'm your host, Ben Shaberman. I'm a science communications advisor for Hope and Focus, and I'm delighted to have Dr. Jason Comander from Mass Eye and Ear join us for this episode. And we're going to talk all about clinical trials for emerging therapies, among other topics relevant to the LCA community. And one thing I want to say before we get started is a little bit about Jason. He's not only one of the world's leading experts in the inherited retinal disease research space, but he also brings incredible passion to his role. And I think you're going to sense that as we go on through our conversation. So, Jason, welcome to the podcast. It's great to have you.
Thank you. It's so great to be here. And thank you for that very nice introduction. You know, I get embarrassed easily. And so I had a feeling I was going to get embarrassed, but thank you.
Well, I appreciate your excitement. So no problem. No problem at all. And I want to give our listeners a little more background on Jason. At Mass Eye and Ear, he is the Ayub Family Chair in Ophthalmology and the Director of the Inherited Retinal Disorder Service. He's also an Associate Professor of Ophthalmology at Harvard Medical School. And he earned his MD and PhD from Harvard and completed a residency, vitreoretinal surgery fellowship, and IRD training at Mass Eye and Ear. And I think what's really cool and exciting is he has served as a surgeon and lead investigator on several clinical trials, including the first commercial gene therapy procedure for any inherited disease, and that was LUXTURNA for LCA2, RPE65 mutations. That happened in 2018. We'll be talking about that. And then in 2020, he was on the team that delivered the first CRISPR-Cas9 gene editing therapy to the human body. And that was a gene editing therapy for CEP290 or LCA10 that was developed by Editas. So two very important, groundbreaking clinical trials. And again, we're going to talk more about those. But to start off, Jason, you know, I'm always impressed with people when they get a PhD or an MD. I think it takes, well, I know it takes a lot of work to reach those achievements. But you have an MD, a PhD, and you're a surgeon. That just seems like a lot. What inspired you to pursue all of those credentials and take on such important work?
Well, thank you for the question. As you know, I don't, necessarily gravitate towards talking about myself, but I guess maybe if the story inspires some younger doctors or scientists to dedicate their lives to helping, people with rare diseases, then it's okay. And I can suffer my own embarrassment. So when I was 16, I started working in an ophthalmology lab. Believe it or not, I was lucky to get a lab tour turned into like a part-time internship. And my mentor there, he was a great scientist and inventor. His name was Jean-Marie Perel. He passed away recently. And he was a PhD. And he says, Jason, if you go into medical research, make sure you get your MD also, because I invent all these things and then I have to rely on these MDs to test them for me. And so that stuck with me. And I do love medical research. I love puzzles. I love helping people through solving puzzles. And it's great to be the person that can help translate them. So I did get an MD PhD. And when I started getting an inherited retinal diseases, I called Al McGuire, who's one of my heroes, did the early surgical development and the decades of work on what's now LUXTURNA. And I was like, Dr. McGuire, can I come watch one of your surgeries? And he said yes. And I went to Philadelphia from Boston for no reason other than I was curious. And he was really generous and let me watch one of the clinical trial surgeries. And I watched that and I just said, I have to do this. And I was already interested in IRDs, but that kind of cemented my interest in both in doing a vitreoretinal fellowship and in making this kind of my career and my passion. So very specific reasons why I wanted to do all that. It's to help people, and I'm living the dream now. I'm doing that, and I love it.
Well, I'm glad it's living your dream, but by also being an MD in addition to a PhD and doing surgery, you're helping patients. And obviously, that's so critical to moving therapies forward.
I mean, yeah, some of my friends have great jobs where, like they're running a business or they're in finance or whatever. I'm so lucky, me, my other peers that are doctors and medical researchers, that we get to help people as part of our regular everyday job. So it's such a wonderful thing to have a mission. And, everybody who's listening to this podcast is in some way part of that mission. So this is great.
Well, again, we're delighted that you are part of the mission as well. So to continue on, I know a lot of people probably already have a general sense of what a clinical trial is, but I think it's important that we're all on the same page about what a clinical trial is as we move forward in the discussion and talk about some of the emerging therapies that have been in clinical trials. So give us your definition of a clinical trial. And in addition to that, what do you think is important for patients and families to know who are interested in trials or at least considering getting into a trial?
This isn't the regulatory definition, but the everyday definition of a clinical trial is testing a potential medicine that hasn't been approved yet. So you don't know for sure if it works or it doesn't. And you're using it on people, on real patients, to help determine if it's safe and if it works. So, it's early. So it's like leading edge, right? So the first thing that I tell patients when I'm talking to them about this is that everybody has their own feelings about being on the cutting edge of something. So if you want to be involved with the latest thing, some people love that. So some people have a different risk tolerance where they want it tested and with a bow on it and FDA approved before they would ever personally expose their own body to it. And that's fine too. And it's natural for people and families to fall on one side of that spectrum or the other. And it's okay to be honest with yourself, yeah, we're going to sit this out until it's proven, or I'm going to go around to as many specialists as I can until I can find somebody who will let me in a clinical trial. And those are both okay and good. So that's the main thing. A couple more things. One is that clinical trials are only one opportunity of how you can potentially improve your overall life. So I was just at the FFB Visions conference, and I was so impressed with what people with various levels of vision loss are able to do. And it just reinvigorated me to encourage people to pursue all of the non-medical strategies for improving their lives, from leveling up all the apps they have on their smartphone, learning how to use them for navigation and identification and reading and communication and all that stuff. There's so many people that can do so much. So while you're in pursuit of the medical therapies, I would say just don't ignore the vision rehabilitation strategies too, because that can make just as much of an impact on your life. And put in a plug for that.
Yeah, that's a great point. Especially the past few years, technology has just blossomed so nicely that there are so many great tools that people with low vision can use to do their daily tasks and things that need to be done. And I think it's also important, while obviously there's risk to clinical trials and everybody needs to be aware of that, we need people in clinical trials if we're going to get therapies across the finish line. And a lot of clinical trials in the LCA space involve kids because kids with LCA may be the best candidates for something like an LCA gene therapy. Can you talk about the special considerations that you take on when you're dealing with kids in clinical trials?
Right, right. So it's interesting because you need someone's permission and their consent like legally to participate in a clinical trial to show that they've been fully informed of the risks and potential benefit or potential not benefit. But when it's a kid, they're not legally allowed to vouch for themselves yet. So the parent has to sign all the legal forms, or guardian. And the kid, depending on their age, there's different responsibilities. So if they're like a baby, there's nothing more to be done. But if they're like 8 years old or over, 10 years old or over, they have to agree to going into the clinical trial. They have to assent, is what it says on the paperwork. And this is for the United States anyways. So you can't like drag a 12-year-old into a clinical trial and force them to do it if they don't want to do it. And I think it's kind of a beautiful thing that there's this collaboration between the parents and the kid to like get them their potential treatment or to at least contribute to science and knowledge about potential treatments for their disease. And I find that the parents are usually really level-headed about it. Like they get it as far as the risks and benefits. I think there's more adult patients that I have to spend more time making sure they're thinking about it the right way. The parents of the young kids, they seem like laser focused on just the right things. It's been my experience.
That's great. And do you think the kids who are assenting, as you say, they understand some of the risks, like a subretinal injection or the fact that you're putting this engineered virus in their eye and all that stuff? Do you think they appreciate the risk and how cutting edge that is?
No, I don't think they do. And I don't think the parents do. And I don't think the adults do either. And probably the doctors too. It's just when you're at the cutting edge, sometimes things happen that you weren't expecting. But I mean, we have like over 10 years of experience in here and, at other places, even longer. And so we kind of like have some boundaries on the risk. So when I talk to people about it, I say, the vitrectomy procedure that we need to do to put the gene therapy in, one of the known side effects is a cataract. And depending on the person's age, it may take longer or shorter for them to get a cataract. But we know you're going to get a cataract. You're going to get a cataract in that eye before you get a cataract in the other eye. But the other risks are more rare. So technically, for a subretinal injection for gene therapy, technically we're detaching the retina and part of the retina. So it's possible that it could come off the rest of the way and need more surgery to fix that. And, I can go through X, Y, and Z and the different ones. And I'm saying, like, fortunately, those complications are rare. And so, you can, anytime you have surgery, you can have bleeding, you can have an infection. Fortunately, that's rare. And so, people get that.
But I guess gene therapy and subretinal injections aren't part of our everyday experience or language. So you can talk about it, but obviously it's kind of challenging for parents and kids to truly wrap their head around what that means.
Oh, yeah. Yeah. And, technically, like the patient or the parent and the kid make the decision whether they want to participate in a trial. But because there's this like unequal information between them and me, I also feel a responsibility to not enroll people in a trial if it's clear that it's like not right for them. And I feel the responsibility about taking good care of my patients too. So it's nice at Mass Eye and Ear we have enough time to talk to people where I do feel like they're fully informed as much as somebody can be without having had the experience before, that I'm happy that we get to practice that way and kind of like build a partnership from the beginning. So do our best.
That's so important. I've been in your clinic, and the amount of time that you or Rachel Huckfeld or whomever spend with patients is phenomenal. And I think that's so critical. Whether it's a trial, an examination, or whatever, you really do a great job just being there with the patient on their journey and helping them understand what's going on. So, speaking of journeys, the treatment, the gene therapy for LCA2, the RPE65 gene therapy, LUXTURNA, was in a trial at Children's Hospital of Philadelphia for many years, but then it went commercial. And if I understand correctly, you, Jason, delivered the first commercial dose of what became LUXTURNA. And that was what, in early 2018? What was that like for you? Was there a lot of pressure? I mean, that's a big milestone.
Yes, and I didn't appreciate it at the time, just just like you were saying. Sometimes it's hard to know what you're getting into. So I'll tell you the story. I thought that Al McGuire was going to do the first one because he had been working on it, like I said, for decades. And somehow his institution got wrapped up in red tape, and Penn was not going to be doing the first one after it got approved. And so I asked my wife, Amy, who's also a doctor, I asked her, Amy, should I rearrange my clinic so that Mass Eye and Ear can do the first procedure after it got approved? Like, it's not really the first one because they did a bunch of people during the clinical trials. So scientifically, it's not really, that big a deal. Maybe it's a new batch of stuff, but it's not that big a deal. She's like, Jason, you definitely have to do that. So I listened to my wife and I rescheduled the patients. And all of a sudden, well, there was a hiccup along the way, which I'll get to in a minute. But the end of the story is that my patient was on the news, I was on the news, and the moral of the story is always listen to your wife.
That's great.
Can I tell you the middle part of the story, though?
Of course.
So you're talking about being under pressure. Everything was lined up. It was so difficult to get this all worked out because this was a totally new thing. Like a gene therapy hadn't been approved by the FDA before. So there's all these workflows and processes that didn't exist yet. And for us to get the drug, they had to ship it to us, but the specialty pharmacy had to ship it to us, but who was going to pay for it and how in advance, or there's 1,000 things to take care of. And the insurance company approved it, thankfully. The patient could travel from out of state. We got the OR time. We got the specially trained pharmacist. It was all ready to go for Wednesday. And so then I get a call from the medical director of the insurance company on Monday. And he said, there's a problem with the approval. And I said, well, you approved it already. He said, well, there's a problem with the genetic testing report. It says that there's a variant of unknown significance there. I said, well, if you read the report, it was written before all this terminology got settled out because it's an old report. And it says there that it's likely mutation. And he said, no, it says in another sentence that it's a variant of unknown significance. And we went back and forth for a while. He said, well, I'm sorry, we can't ship you the drug. And I was thinking okay, they got to call the patient, got to call the TV cameras. I was like, well, just give us some time. Let me see if we can get some more information on this. He said, okay, you've got 1 hour until the shipping deadline. So I went to the genetic counselor. Actually, it was a genetic counselor assistant because the genetic counselor was out. And I was like, we got to do something about this genetic report. And for those of you that do genetics, it usually takes months or years to get your genetics settled. But within the hour, we found another family who had the same mutation and published in the medical literature. Actually, I was checking PubMed and the genetic counselor was checking Google, and she found it first. And that just barely met the criteria for it to be acceptable to treat. So within that hour, we faxed a new report back to the insurance company. They sent us the drug, and the rest was history, and we were on the news and went really well.
That's so crazy. They gave you 1 hour
Mm-hmm.
and you go out on PubMed and you find another case
Mm-hmm.
and you make it all happen. That sounds so unlikely.
I know. And I had already been interested in this before, but it's even more motivation for the part of my research laboratory where we're doing assays on different gene mutations to see which ones break the gene. And so that less people will have variant of unknown significance on their genetic testing report. So it's good. It's good motivation that's a real life problem. And the story totally exemplifies that. Before that, I thought it was kind of silly or not important to be on the news or get any media or anything. But it actually led to some important good things. I could be on this insurance panel to help get LUXTURNA approved for people and stuff like that. So yeah, it was a good thing. Listen to your wife.
Good advice. Good advice.
Mm-hmm.
And I didn't realize that you were kind of plan B for Al McGuire because, as you pointed out, Al did all the surgeries in the clinical trial. It's kind of funny that he didn't have the clearance or whatever, or the approval to do the actual injection of the commercial therapy. So
I was plan B in my mind, as far as I was just assuming that it would be fitting and proper for him to do it since, it was his baby for so long. And Gene Bennett, those are my two heroes, the husband and wife team who developed this. And by the way, it should be said, to push this over the line and get it into people and get it to where it is today, they decided to give up all financial rights to LUXTURNA, and the company was sold for $1 billion. And so they were very selfless in doing that for the LCA community and for the entire field of inherited retinal diseases and gene therapy. So that's only one of the many reasons why they're heroes.
That's a really great point because Spark Therapeutics, the ultimate company that commercialized the therapy, they were sold to Roche for like $4 billion.
And my experience with LUXTURNA since that day when we did the first one has really been amazing and a lot of fun. So I have lots of good stories from my patients. And the main thing that LUXTURNA does for a decent fraction of patients who have enough anatomy to support it, it gives them night vision. And I try not to say gives them back night vision because it gives them night vision that they've never had before, which is incredible. And so I do the surgery. I'm looking at the eye afterwards. And, at the week 1 post-op, it looks like nothing happened. I mean, if you look carefully, you can see there's no vitreous there, but usually we're peeling off some scar tissue or fixing an attachment or whatever, but the eye looks the same. And yet they come back in a month and they're like, Doc, I can see my clock on my nightstand. I've never been able to see that before with the lights off and things like that. I have a PE teacher who said before the surgery, the street lights on my street, they lit up about 10 feet, a circle of 10 feet. But after the surgery, they lit up 30 feet because he could see all that dimmer light. And he said it helped him at night games and that kind of thing. And one that I'll never forget was this 3-year-old girl who got LUXTURNA. And it was really difficult to get it all done. But in the end, she was there for her 1-month visit and I was curious if it worked or not. And, the 3-year-old can't do the traditional visual field test to measure that kind of thing so I did the best that I could with what we had, which was I turned the lights in the room off and there was some background glow from the computer monitor and stuff. I took out of her stroller, I picked up her baby doll and I held it up in the air. And as soon as I did that, she grabbed it. And I'm thinking in my mind, well, was it dark enough in here to really make that the test? Would she have been able to see that anyways? And I look over to the mom and the mom is crying. And that's the moment when I knew that we had done something for her. And I was so moved and so touched. And LUXTURNA is great. I hope I get to do many, not that I want anybody to have a problem, but for those that do, I really love doing the LUXTURNA for people.
Right, right. Thanks for sharing those wonderful anecdotes. Those are very moving. And I'll add just briefly, because I do want to talk about the Editas SEP290 surgery, but we're seeing those kinds of results with other LCA gene therapies, like it's seen as GUCY2D for LCA1, LCA5, even LCA4, which is really severe. Those gene therapies are all restoring vision. So LUXTURNA really is leading the way to hopefully some more approvals in the not too distant future. But let's move on to the Editas CRISPR-Cas9 trial. And I guess quickly, can you talk about what the difference between that approach is versus LUXTURNA or the gene augmentation, gene replacement therapies that I think are more common or we're more familiar with?
The terminology all sounds complicated, but the concept is actually really not complicated at all. So the genes are little bits of instructions that make protein machines in your body. And if the gene is broken, the protein machine is not working, and there's, the cells in your retina are not working right. So one kind of straightforward way to address that is you take a correct copy of the gene and you put it back in the retinal cells. And that correct copy makes the protein that's missing. No problem. But the way that we get the correct copy back in there is we use a specially modified virus that delivers the DNA into the cells. And that virus has a top limit of capacity that you can only fit so much DNA in it. You can try to use a different virus that has a little more and doesn't work as well, etc. But basically, once the gene becomes a certain size, there's no easy way to get a fresh copy of the DNA in there. So one alternate strategy is just to fix the DNA that's already in the cell. Now it's broken. That's the thing. And so you can't make it in the lab and purify it and put it in. You have to work with what's in the cell. And this was pretty much impossible to do it with any reasonable amount of efficiency until recently when CRISPR-Cas9 came along. And the original Cas9 is like little molecular scissors where it'll find a particular place and all of the DNA in your cell and chop it up. So there's only certain mutations where that's going to be helpful. But it just so happens that in CEP290, there's one particular DNA change that you can kind of like chop out the bad area and the gene will start working again. And that was this particular mutation in the CEP290 gene. And so Eric Pierce, who's another one of my heroes, who's the director of the Ocular Genomics Institute here at Mass Eye and Ear, he convinced Editas Medicine to design their first in vivo, in human gene editing clinical trial to be for this CEP290 mutation. Now, in the past, the Cas9 had been used, CRISPR had been used by taking cells out of the body, fixing them, make sure they're okay, take the good ones, put the good ones back in. And they did this for blood cells. But for the retina, it's a whole different story. You can't take the retina out and fix it and put it back in because it's connected to the optic nerve, to the brain. So you got to fix it in place. So this was a big deal because now the CRISPR tools were being injected directly into the person's body. And this was, at the time, it was the first time this has been done. So talk about pressure. I had no idea what was going to happen once we injected this stuff into people. So you kind of know because you've done extensive safety studies. It's been done on all kinds of animal models. And you kind of feel, like it's supposed to be okay, but there's still something in your head, in my head, that said, gee, I really hope this is going to be okay. And I don't know what it's going to be when I take the patch off on the first day. And it was okay. It was okay. It went like other gene therapy programs, not very much different as far as, all the different considerations and things that can happen. And you know what? It worked for a fraction of people. There was a fraction of people who saw better. And that was so cool because not only, most importantly, we're helping those people see better, but second, we've shown that this new technology of taking this crazy DNA editing machinery that was originally found in some lower animal archaea, modified it and using this very crafty way, using it in people and showing that we can actually fix the DNA that's already in the person's cells without taking it out and manipulating it. It was quite the demonstration that this gene editing technology works. And that was super cool.
Right. And I think an important lesson there is ultimately Editas did not take this gene editing therapy forward. Like you said, it worked for some people, but not in a big way for a large group of people. So they decided not to move it forward. But it did show that you could do it safely and that it did have potential to work. So it still moved the field forward, the gene editing field forward. Would you agree with that?
I agree that it moved the field forward, but I have to say that I was disappointed when I saw the world's reaction to what I thought was pretty amazing. So this is a very difficult problem where somebody buried in their 3 billion pairs of DNA, they have one that's broken. There's no way to get a complete copy of the gene in there. And we were able to go in there and fix that one spot in the person's DNA, and a fraction of people saw better. That's crazy. And so the fact that it wasn't 100% of people, I just thought that was too high a bar for the situation. And the reality, I think, was why they're not sponsoring a phase 3 trial is there's not a big patient population. So at the beginning, there was interest in it from multiple perspectives, but as time went on, the interest, waned when it wasn't a big market. And so, a lot of the doctors and thought leaders in the IRD field are planning how down the road we can not leave anybody behind and how we can address all of the people that are in, smaller groups of people, and a lot of LCA people fall into that.
Sure. That's a very important point, is to help these smaller populations and people with specific mutations that are amenable to gene editing. And we are seeing more gene editing therapies move toward clinical trials for IRDs and so on. So the field is moving forward. Would you agree with that?
Yes. The things that move forward the easiest are the ones that have enough population where a company is going to put enough money into it to do a decent sized clinical trial. But there are signs from the FDA that they're willing to be more flexible and that there might be more than one pathway to get to a treated patient than, what's required for, the traditional route.
Certainly. And you see, at least with traditional gene therapy, where putting in a whole new gene. Conditions like LCA5 and LCA4, which are really rare, that companies and the regulators are finding ways to move those forward.
Yeah, LCA4, that's a great story. So the initial early human clinical trials were so impressive where they treated these babies, and it wasn't like you needed a lot of statistics to figure out the results. Like the treated eye could see and the untreated eye could not see. And they showed the pictures, like you could see the cone, the line representing the cone light sensor cells in the treated eye, and there were none of those in the untreated eye. And so that's amazing. And so Lilly licensed that from Myra GTX, and we're hoping that we're going to be one of the sites that gets to treat kids with LCA4.
Oh, that would be very cool. Very cool. We'll stay tuned. So to close out our discussion, I know we've been talking about gene therapy a lot, gene editing somewhat. What are some of the other technologies, the therapeutic approaches that you see coming down the pike that you're excited about, especially for the LCA community?
Right. So, like I was, we had both mentioned the AIPL1, the LCA4, one of the more severe ones. And so there's a lot of people that don't have the rod and cone cells to put the gene back into, right? So what do we do for those folks to not leave anyone behind? Like I was saying, well, one approach that I'm most excited about for that is optogenetics, where, okay, so you don't have any cones or any rods to put the original gene back into, but what if you take the remaining parts of the retina and make those light sensitive? So it's a type of gene therapy where we're delivering a gene, but we're delivering an artificial light sensor to the remaining parts of the retina. And those parts weren't originally intended to be light sensing, but they're all connected up to the brain and such. So it's possible that you could restore a decent amount of vision by using kind of like hacking in the light sensors into the remaining cells. And a number of companies have tried this, and I think the technology is evolving, and we're going to hopefully be getting a trial here at Mass Eye and Ear for that pretty soon too. And it's taking some time, but I have a good feeling that approach is eventually going to work.
Right, right. There's a lot of trials right now for people with RP, but the same approach can apply to people with LCA because it's the same scenario. As you said, they've just lost all their rods and cones. You're harnessing other cells in the retina for vision.
I have something controversial to say. I hope nobody gets mad at me. LCA is pretty much just a severe form of retinitis pigmentosa. So when we were doing the CEP290 trial, they're supposed to have LCA, but when people came in to be screened for the trial, there was people who had like regular old RP, and there were some that even had decent vision, like into the like 30s and 40s, and CEP290 patients are not supposed to have that. So, patients come in to my clinic and sometimes they get confused, like, oh, this doctor said I had RP, this one said I had LCA. So if you're young and you have nystagmus, meaning the moving eyes, you're more likely to be called LCA. And if you're older and you don't have that, you're more likely to be called RP. But I think like these technologies are going to be able to cross those lines really easy.
Right. I don't think that's controversial. I think that's a really important point that the LCA term, the RP term, they're just labels. At the end of the day, somebody has genetic mutation or mutations and their retina is affected in a certain way, and you're just trying to address the condition. So I think that was a really good point. The very last thing I want to ask you about are cell therapies, where you're actually trying to put in some new rods and cones. So cell therapies haven't advanced as aggressively as gene therapies. Where do you think we are in cell therapy development right now?
I think it's earlier days for cell therapy. So at Mass Eye and Ear, we did a cell therapy trial starting in about 2017, where we were implanting subretinal retinal progenitor cells. And there were some people who we thought might have benefited, but overall, my conclusion is that I don't think it worked. And I haven't been, and it, anytime you do surgery on somebody, there's some fraction of people who have some issue or some problem. And right now, I feel like the technology is not quite there yet. And I'm focusing on the gene therapies and other technology. And I've, declined to participate in some stem cell technology since that time. It doesn't mean it's not going to work. And it doesn't mean that it shouldn't be tried, but it's just, I'm less bullish on that. But I should also say that there's two approaches for the stem cell technologies. One is to put the cells where they're supposed to be and hope that they connect up to the retina and actually act like a rod or a cone light sensing cell. There's another approach that a company J-Side is doing where you put some stem cells into the middle of the eye, into the vitreous, and they're supposed to secrete factors that are good for the health of the rest of the retina. And so they're not really being asked to connect up to the rest of the retina and act like, true rod and cone cells. But on the other hand, the mechanism of how it's supposed to work is like one more step indirect, that they're being asked to secrete something that helps the retina that's already like not healthy. So I hope all of these things work. But if you're asking me, I'm personally spending more time on the gene therapy based approaches.
Right. Fair enough. Fair enough. And it's important to have all these modalities in development because ultimately some are going to work better than others, and some are going to be better for certain patients and others will be better for a different group of patients. So
yeah,
it's important to have all these things in the pipeline.
And there's also the microchips are showing some more promise now too. So there's a Primavera Trial Science Corporation. They're implanting microchips, straight up silicon under the retina. And they're doing it initially in age-related macular degeneration, again, a big market. And they're showing some really interesting results with that. And I think it's going to be tested in IRD patients eventually. I have a good feeling that it will be. So
Right. I know people with that company have an interest in the inherited retinal disease space. So hopefully they can translate that over to IRDs.
I would like that. Yeah.
Yeah. Jason, it's been a lot of fun to talk to you, to learn about your experiences on some of these really groundbreaking trials, to hear about your wife Amy's influence. We'll give her a nod. And I'm just curious, now that I'm mentioning her, what kind of medicine does she practice?
So Amy Comander is a breast oncologist, and her specialty is lifestyle medicine and survivorship. And she is way more famous than me, and I think she's amazing.
Well, you're being very modest. Clearly, you're both amazing and doing very important things for the health and well-being of people. So thank you.
Well, thank you. And I hope the stories and vignettes in our discussion here really fulfilled the purpose of bringing hope into focus for all the LCA patients.
Well, that's so well put. Well, this discussion has given all of us a lot of real hope. So thank you again, Jason. And thank you to Eve Orcutt for helping with the recording and production. And listeners, thank you as always for joining the Hope and Focus podcast. It's great to have you, and we look forward to having you back for the next episode.
Whether you live with vision loss or love someone who does, this space is for you. To learn more about Hope and Focus and join us in advancing treatments for LCA, visit hopeandfocus.org and connect with us on social media @HopeandFocus1. Together, we're bringing hope into focus.